In this video, we will study about monohybrid cross. It is a cross between the organisms of a species which is made to study the inheritance of a single pair of genes or factors of a character. Now, in this monohybrid cross, we will consider one character at a time. Hybrid mean we are going to make a hybrid by cross breeding, and at a time we will consider single character. So this is called as monohybrid cross. Now for this cross, we can select certain characters like plant height. This may be tall, this may be dwarf. One is dominant character, second is recessive character. Second is flower and pod position. It can be in the axis of the leaf, axial. It can be at the tip. It is called as terminal. Now this is represented by capital A and small a. So this is a dominant gene. This is a recessive gene. Similarly, color of the pod. It may be green. It may be yellow. Capital G is dominant. Small g is recessive. Then pod shape. It can be inflated shape. Capital I, constricted, small I. Now shape of the seed, it can be rounded, it can be showing wrinkles, small r. Color of the cotyledon in the seed, it can be yellow, capital Y, it can be green, small y. Then color of the seed covering, seed coat, and flower color may be colored, and this may be white. So these are the characters being determined by factors or genes. Now these factors or genes, they are located on the chromosome. See, supposing this is the factor or gene which is controlling a particular character. Now this may be dominant, this may be recessive. So for monohybrid cross, we will take one character at a time. Supposing we are taking plant height of the pea plant. Now we can take tall variety and we can cross with dwarf variety. We can take rounded seeds, we can take wrinkled seeds. Similarly, we have characterly discussed in the earlier slide, axial, terminal, then flower colored, flower white. We can take any character at one time. So one character being considered at one time is called as monohybrid cross. Now in this cross, we will take tall plant and dwarf plant. One may be female, second may be male, and vice versa. Now this will produce gametes with capital T genes, and this will produce gametes with small t genes. Now this is homozygous dominant, this is homozygous recessive. So they are producing gametes which are very pure for a particular character. This will have either capital T, this will have small t. And when they are being cross fertilization is occurring. So when we make a Punnett square checkerboard, capital T will combine with small t, capital T will combine with small t. Now this will be capital T, small t, capital T, small t. Now this capital T will combine with this small t. This will be capital T, small t. Capital T will combine with small t. This will be capital T, small t. Now this capital T and small t, they will form a tall variety, but this is not now homozygous like this. This is heterozygous, or we can say this is a hybrid between the two parents. So 100% first filial generation, F1 generation means first filial generation, will be tall, but they are hybrid in nature, they are heterozygous in nature. Now, second, we can consider self-breeding of the F1 generation. So what is self-breeding? Now this is the flower of the pea plant. Now this is pea plant flower. Now it has Papillonaceous corolla. What is Papillonaceous corolla? Now we can see this is the big petal which is present toward the outer side, and these are two lateral petals, and this is the here two petals which are fusing to form a boat-like structure called as keel or carina. Now if we open the flower, we can see keel, this is the keel part. Then these are wings, and this is the Papillonaceous Corolla. Now we can see here these are the stamens, fused stamens, and this is the ovary style and the stigma. So this is ovary, style and stigma, and we have pollen grains. No. Now, if it's pollen grains, they fall on the same stigma. This is called as self-pollination or self-breeding. So when in case of pea flower, flower may not open up and self-pollination may take place. In case of pea flower, if we want to do cross breeding, that is, if we have, this is ovary, style and stigma. These are its stamens. We have another flower. Now this is the ovary, style and stigma of second flower. This is A flower. This is the B flower. These are stamens. Now if pollen grain of this A flower will fall on the stigma of the B flower, this is called as cross breeding, and this in case of pea flower we do it artificially. So now we will take this F1 generation hybrid tall plants and we can proceed for its self breeding. So this is self breeding of F1 generation or first filial generation. Now in this case we are going to take plants which are heterozygous hybrid tall. This is female, this is male, and these are parents. Now they will form gametes. This will where female will have egg cell and male will produce pollen grains. They will form male gamete. Ultimately fertilization will take place. Now because here this is heterozygous, so this will form two types of gametes, one with capital T and second with small t. Though they are hybrid tall, but gametes, they are always pure for a particular character. They will have either capital T or small t. Here also this will produce the pollen grains with capital T or small t. Now when this capital T will combine with capital T and small t will combine with small t, capital T will combine with small t also. So in this case here on one side we can show male gamete, another side we can show female gamete, and we can represent it by Punnett square method. So capital T will combine with capital T, so this is pure tall. Capital T will combine with small t, this is hybrid tall. Small t will combine with capital T, this is hybrid tall. Small t will combine with small t, so this is pure dwarf. Now these two parents offspring, they are homozygous and these are heterozygous, so they are hybrid in nature. So we can see here gametes, they are segregating independently and they are pure for particular character. Now this generation is called as second filial generation. In this case we can see we have 75% tall, which will be having 25% pure and 50% hybrid, and we have 25% pure dwarf. Now phenotypic ratio, phenotypic is what we observe externally morphologically, this is called as phenotypic ratio. Now we can have 3 tall and 1 dwarf, so ratio will be 3:1. Now if we see its genetic character, genes genotypic ratio, we can see this is pure homozygous, so this is pure tall, this is 25%. Now this is capital T small t, capital T small t, those they are forming 50% of hybrid tall, capital T small t. Their gene, though this is tall, but this is homozygous, this is heterozygous. Now this is pure dwarf having small t genes, so this is 25%. Now ratio, if we see its genes or genotypic ratio we call it, this is 1:2:1. So in monohybrid cross we will consider single character at a time. Now its phenotypic ratio is 3:1. Its genotypic ratio is 1:2:1. Thank you for watching my video. If you like my video, please like, share and subscribe.